Good evening, all. Welcome. What a great crowd. To our chemistry students who are everywhere, does chemistry rock? Okay. And there'll be a quiz afterwards. That's a joke, a bad joke, but it's a good joke. So this is one of my favorite moments and one of the great moments in the life of our college or any institution of higher learning where we have an opportunity to celebrate a singular honor that's bestowed on a member of our faculty to recognize their amazing work and contribution intellectually and in the classroom on a consistent basis over a number of years. We have many deserving faculty who have received an endowed chair, none more deserving than Dharni Vasudevan. So, Dharni, congratulations. So this is the inaugural lecture for the Stanley F. Druckenmiller Professorship of Chemistry and Environmental Studies. This was a chair that was endowed by Stan Druckenmiller, class of '75. I suspect many of you know Stan's name and his history. Some may not, but a legendary investor, one of the most successful investors ever, and an incredible philanthropist, incredibly important to Bowdoin College and the modern Bowdoin College in so many ways, and incredibly important to so many other great institutions in our country in the life sciences and education in the work to fight poverty. I'm privileged to have gotten to know Stan well, and I know that he is here in spirit with us tonight and would celebrate greatly your receiving this chair, Dharni. Now, Dharni arrived here in 2004, jointly appointed in chemistry and environmental studies. She is an exemplar both of the interdisciplinary work and of the collaboration with students and other faculty members across these disciplines and encroaching into others. Her research focuses on the ways that pharmaceuticals, pesticides, and other chemicals affect our soil and our aquatic environment. Her work's been supported by the National Science Foundation, Merck, AAAS Undergraduate Science Program, the U.S. Department of Agriculture, and others. And she has a devoted team of student researchers in her lab who refer to themselves as Team Dirt. She has published widely in peer-reviewed journals such as Environmental Science and Technology, Chemosphere, and Geoderma, very often with students as co-authors. She's given nearly 100 talks at conferences and other colleges and universities and institutions, again, often with her students as co-presenters. She spent the academic year 2013-14 as a visiting science in the Exposure, Epidemiology, and Risk program at the Harvard School of Public Health. She was featured in a documentary about humans' relationships with the natural world called 13 Ways, which premiered in the Environmental Film Festival in Washington, D.C. in 2019. Dharni, because that wasn't enough to keep her busy, also serves as our Associate Dean for Academic Affairs for Faculty Development and Inclusion, and I've had the chance to work closely with Dharni in this role. I've come to admire her amazing impact on this college from an angle different and from her teaching and her scholarship, but equally important for the benefit of our college and for our future. She has really played a key role in the DEI work that is going on, not just within academic affairs, but across the college more broadly, and for a time teamed with Benji to carry our larger campus-wide DE effort through a very challenging moment. She received her undergraduate degree from MIT. We spent some time talking about that tonight. Her master's and PhD from Johns Hopkins. She is an influential, accomplished, collaborative scholar whose work has urgent implications for our natural environment. And as Team Dirt will tell you, she is an amazing mentor. I'm delighted to introduce tonight our Stanley F. Druckenmiller Professor of Chemistry and Environmental Science. Please join me in congratulating and welcome to the podium, Dharni Vasudevan.
Thank you, Clayton. Thank you so much. Thank you. Situated. So thank you, President Rose, for that generous and kind introduction. Thank you, Dean Scanlon, for your words from a little while ago this evening. And my sincere thank you to Mr. Stan Druckenmiller for endowing this professorship in environmental studies, which I'm honored and humbled to be a recipient of. I'm so happy and grateful for everyone who is here today in Kresge and joining via live stream. And for those of you joining from India, it's 5 a.m. I'm sorry, but I'm really happy that you're here today. To my parents, Amma and Appa, your belief in the transformative power of education, freedom, and kindness has me standing here today. Thank you. So I'll be talking about chemicals in the environment, tales of attraction, repulsion, and fate. And before I do, I want to acknowledge my co-authors, my co-creators, my co-weavers of these tales, our Bowdoin students. The brilliant, hardworking, insightful Team Dirt crew that I've been fortunate and honored to work alongside. Most people you will see on this slide have worked and contributed to the work I will share with today. A few who were not directly involved with the subject of today's talk were important partners asking insightful questions and pushing our analysis forward. So in somewhat chronological order, Gia, Amber, Anthony, Brendan, Tim, Liz, and Kelly, Mark, Mo, Adam, Teresa, Tina, Andrew, Ali, Amy, Hadrian, Basil, Daniel, Phoebe, Rhea, James, Alondra, Katie, Malik, John, and Jillian, Danielle, Leah, Sam, Jorge, Emma, Eric, Tyler, Chiamaka, Xander, Ronmel, and most recently Ben, Chess, Seamus, and Mariah. So it's really been a privilege to work alongside these students. A few of them are in the audience today, and thank you for those trying to join live stream. And another really important person in this journey is Allison Mckay, my long-term collaborator for almost 20 years. She's now at Ohio State University, and when we started, she was at the University of Connecticut. Most recently, her student, Will Jolin, we worked with him pretty closely too. There are a few people at Duke University who were part of this effort for about a year when it began. Ellen, Aaron, and Sheila, and Greg Bruland, who continued to help with the statistical work. Jose Gascon and his students at Connecticut, who helped with some of the computational modeling. Back at Bowdoin, Paris Trivedi was a postdoc in my lab for about a year, and Celeste Morin, who keeps our instruments healthy and my students and me happy. And Martha Black, more recently, has really partnered in helping me as I spend more time at Druck. So I'm really grateful for all these people who I got to create these stories with. And importantly, I would like to acknowledge the funders that we've been fortunate to have funding from the National Science Foundation, the USDA, Bowdoin College, and the many sources of student fellowship funds. So I'm really grateful to have that. So today we hold this difficult duality of life-sustaining and life-endangering properties of chemicals. So many of us in this room have and do rely on pharmaceuticals for our health and well-being, and particularly in the last couple of days, on waterproof, windproof, breathable Gore-Tex. And we hold this reality that we use chemicals, but these chemicals get out and move. And we have evidence of these chemicals getting out and moving as we hear stories about PFAS getting to polar bears, I had to get started on that one. Farmed salmon containing contaminants, and arsenic and lead leaching out of fertilizers. So we use chemicals, and they get out and move. We need them because they're life-sustaining, but at times they can cause problems and be life-endangering. So here are some examples. Downstream of a wastewater downstream of a pharmaceutical plant, you see more development of intersex or characteristics of both sexes in fish, increase in antibiotic-resistant infections, and male bass in the Potomac producing eggs. So we live this reality that we use chemicals, they get out and move, and they can cause problems. At the same time, we are balancing this duality of life-sustaining and life-endangering aspects of chemicals. So if we think about how might problems arise and how might we be at risk, we can think of risk as being determined by a combination of hazard and exposure. So in this case, exposure to the sun, for example, and the hazard could be a range from just overheating to a light sunburn to more dramatic exposure to UV rays. And with respect to chemicals exposure, very much what we are exposed to depends on how and how much we ingest chemicals through the air we breathe, through the food products or water that we consume. And if we think about hazard or harm, it's determined by the effect of the chemical has on an ecosystem or humans, be it toxicity, carcinogenicity, or neurological effects. So essentially, we can limit exposure. We can limit harm if we limit exposure. And so an example is with and without an umbrella. So you're limiting exposure, we limit harm. And a parallel in the case of chemicals would be to limit exposure to the chemicals, then you limit the risk and you limit the harm. As this slide characterizes, we have a long way to go with respect to characterization of exposure and hazard of chemicals. Of the 100,000 chemicals in the market, there's only about 1,100 chemicals that are well characterized for both exposure and hazard. And so if we think about exposure is determined by chemical fate, which in turn drives the chemical's personality. So what Team DIRT gets excited about is the exposure side of this, is the idea that we can discover and anticipate a chemical's fate in the environment if we have a window from its structure into its personality and then the behavior of the chemical. So take ciprofloxacin that I've shown here. It's made up of a lot of carbons and hydrogens, but you can't see them, and more on that later, but you see oxygens and hydrogens. And if you think about the bonding arrangement, there's a specific bonding arrangement to ciprofloxacin, and this defines its internal energy of this molecule. Now, if this ciprofloxacin is in a tablet or a jar in our lab, it has a certain surroundings. It's surrounded by other ciprofloxacin molecules. If that ciprofloxacin is now in water, its surroundings have changed and it's surrounded by water molecules. If that ciprofloxacin is in soil, it's surrounded by various soil components. Now, what the molecule is surrounded by also drives its external energy. And so if we think about this internal, external energy, they're both driven by the structure and the surroundings of the chemical, which in turn influence chemical behavior and its personality. So and it also determines what the chemical is attracted to or repulsed by. And so how long the chemical stays in a particular environment, whether it moves out, comes back in, or goes back and forth, all depend on this combination of internal energy and external and its surroundings. And so two examples of this would be a chemical released in the equator can grasshopper its way up to the poles, or you could recall how PFAS released in one area can move up into polar bears. And so we can think about how then does the structure influence the behavior of the compound because of its own set of internal energy and its interactions with the surroundings. Perhaps there's some similarity to us, so it's a little bit of a stretch, but I'll try. Our behavior is also driven by our internal energy, whether we've had a rested night or we're tired, and also our surroundings, whether we're indoors or outdoors, whether stuck in a traffic jam, being late to an appointment, or whether you're watching a sunset with your friends. And so our behavior, what people and situations we are attracted to or repulsed by, also is determined by our surroundings and our internal energy. So chemicals are a little bit like us sometimes. So coming back to chemicals at large, chemicals can move between Earth system compartments, between the air and water, and vice versa. They can also move from the water onto sediments and into soils. So our work over the last 18-ish years at Bowdoin is to understand what drives pharmaceuticals, pesticides, and related chemicals in soil water systems. Specifically, we've asked, what is the driving force that moves a chemical from water into soils, and what role does attraction and repulsion play in this? So the tales you'll hear today are about pharmaceuticals and related compounds in soil water systems. Specifically, we focus on a process called sorption, which accounts for absorption into the solid and adsorption on the figure. So I'll use the term sorption a lot, which is a catch-all for both those terms. So to set the stage for the tales of attraction, repulsion, and fate, a little bit about chemical structure. So an organic chemical is a compound with at least one carbon-hydrogen bond, usually a lot more carbon-carbon bonds. And think of a bond as a net attraction between the net attraction that results, say, between two elements that results from the attraction of the positively charged nucleus to the electron cloud, but balanced out by the repulsion between two electron clouds. And so if we think about chemicals here, carbon is really unique in that it can have 4 bonds. And I show you the structures of glucose, isopentane, which is found in gasoline, and ciprofloxacin again. And a little bit about the notations that lazy chemists use. So a dash is just a carbon-carbon bond. And since carbon has 4 bonds, if you don't see 4 bonds to a carbon, that means there are hydrogens that you can't see. And why is that? So if you look, maybe we can tolerate all the carbons and shown on glucose and on isopentane, but this is not so pleasing to the eye. And so leaving out the carbons and assuming that the dashes are carbons allows one to visualize these chemicals much more. So I hope I've demystified organic structure a little bit for you. Lots of dashes means lots of carbons and hydrogen bonds. And if there's any other element like an oxygen or nitrogen or sulfur, you'll see it on the molecule. So early on, environmental chemists were focused on legacy chemicals like DDT that Rachel Carson wrote about, the eggshell thinning of DDT, and the fear that the world would wake to a silent spring from the loss of birds. This group of chemicals included compounds like PCBs, chemicals in soot, and other pesticides. And these compounds were primarily made up of carbon-carbon and carbon-hydrogen and some chlorines bonded to double-bonded carbons. Here is the structure of water, very different from these chemicals. And these legacy chemicals tended to be water-hating or hydrophobic and were driven out of water into soils or organisms. And environmental chemists have a pretty good understanding of the fate and transport of these chemicals. With time, we began detecting these other chemicals in the environment, both because we were producing them and using them, but also because we were able to detect them better. Our instrumentation was doing better and better, and so we could detect them in the air, the water, and in organisms. And this really allowed us to see what was going on with these new sets of chemicals. But these chemicals were less water-hating. They stayed in the water a little bit more. And why were they less water-hating? So we can go back to the structure of water. We think of water as made of oxygen and hydrogens. However, oxygen and hydrogen have very different electron attracting powers. So the nucleus of oxygen can pull the electrons in the bond much more towards itself, and this pileup of electrons creates a kind of partial negative charge on the water. Now hydrogen is left with a deficit of electrons and has a partial negative charge. So coming back to these compounds being water, somewhat more water-liking, is because the carbon-oxygen and the carbon-nitrogen bonds, there's again a difference in electron attracting power, which creates partial charges in parts of the molecule. And the idea is that compounds with opposite partial charges are considered polar. So the partial charges on these molecules can be attracted to the opposite partial charges of water and make them more soluble. So I couldn't resist some polar bear humor. So which one of these bears will dissolve in water? Obviously the polar one. So what makes a compound even more water-loving is if it has a full-on charge and not a partial charge. So here are some examples of pharmaceuticals which have the potential to ionize and carry a positive or a negative charge. And it's easy to figure out where these charges might be, because if you have a group like a COOH group or OH group present, that H can be lost and a negative charge can be created, or whenever you see an N, there's a chance that you might get a positive charge on the compound. So essentially, pharmaceuticals can be positive, negative, or neutral, or have both charges present on them. And so the takeaway here is lots of dashes, not much charge. If you have presence of N, O, or S on the molecule, it make it a little bit polar with partial charges. It might like being in water a little bit better. And if it has a full-on charge, it's going to really like being in water because it can be attracted to the opposite charges on water and allows it to stay in water. So then now a little bit about soils, the life-supporting skin of the earth. Here we have the soil profile from the organic layer down to the bedrock. And soils are comprised of organic matter. And this organic matter is the black top that you see on your soils. It's basically comprised of dead leaves and litter that has been decomposed over time, perhaps a dead possum that has been decomposed over time by microorganisms. And so we can think of soils as having this organic matter and then the mineral content, so the mineral part of the soil, which is not carbon-based. And we have the microbes in there that do the decomposition that keeps the soil alive. Now, when we come back to thinking about the context of chemical fate, there are three components that are really important to the fate of a chemical. And these include organic matter, which I talked about, aluminosilicates made up of aluminum and silica, and metal oxides, usually of iron and aluminum. You can see that most of these components can carry a negative charge, and there's some smaller amount of positive charge. But soil is not really net charged. Typically, what you have is naturally occurring elements with an opposite charge, like shown here, calcium, sodium, potassium, that sort of neutralize this charge. So the takeaway here is while soil is net neutral, soil has some charges within it, and this will become important when we think about attraction and repulsion and the idea of opposite charges attracting and like charges repelling. So we have our molecule with charges and we have soils with charges. So what Team Dirt studies is the driving force of a chemical to move from water to soil, or this process called sorption. So consider a solution with the negatively charged component and the neutral component of a set of molecules that are now enter the soil profile. When it goes through the organic matter phase, since organic matter has a negative charge, then the negative molecules are going to stay in solution because they're repulsed by the organic matter, but the neutral compound might get bound to it. As it enters the mineral soil, The negatively charged compound could now also bind if there's an attractive force. So soil acts as a filter, and very little of the compound goes into the groundwater. In a different way, we can think about this as absorption process, where the chemical, which is in water, moves from water onto the soil, right? So this is the retention process that we study. We have to measure it and decide on how much. And so we measure a constant called KD, which is the equilibrium constant, which just tells you how much got on the soil and how much stayed in water. So if you have a large, KD is a large number, then you have more sorption, so more went on the soil. And if KD is a small number, you didn't have as much sorption on the soil. So you'll hear about this, the term KD a little bit later, and it's just an actual number that measures how much got on the soil. So the tale begins. Once upon a time, in the 1990s and early 2000s, we began detecting super low concentrations of pharmaceuticals in the environment. However, our theories and mechanistic understanding we had for organic sorption were largely based on those water-hating legacy chemicals like DDT, which were not working for pharmaceuticals. We were unable to predict or anticipate what pharmaceuticals could do in the environment. Allison Mckay and I had studied different molecules in different systems for our PhDs, and were in the early stages of our career. We met at a conference where we were discussing how these theories were not working to anticipate the fate of pharmaceutical compounds. And we decided to bring our complementary training together and write proposals to answer these questions. How can we predict or anticipate, so we don't have to measure all the time, how much pharmaceuticals will sorb onto soil so they could be filtered out? And what are the driving forces then that drive the pharmaceutical from water into the soil? As good, naive scientists at very early stages of our careers, we decided to go with answering the question with the most ridiculously difficult setup we could find, largely ignorance. We focused on the two of the highest used pharmaceuticals, particularly relevant to animal husbandry and factory farming, oxytetracycline and ciprofloxacin. And we selected 30 soils from the eastern and southern United States. And I say ridiculously hard because these compounds have both positive and negative charges, and their charges change with the pH of the surroundings. And so rather than starting with a simple structure, we started with the hardest pharmaceutical structure possible. And for these 30 soils, we measured lots of soil characteristics. Some I'll point out here: surface area, the amount of negative charge, because that's really important, the pH, the amount of organic matter, the amount of surficial and iron and aluminum, so what's on the surface, and other properties. And we measured sorption to soil in very simple lab experiments in which we intentionally contaminated soils with pharmaceuticals. So this was all done in the lab in very controlled experiments, and we measured the sorption constant KD, which tells us how much sorption can occur. And instead of telling you how exactly we did the experiments, I'll just show you some pretty pictures of students and I doing these experiments. We did analysis. We washed lots and lots of dishes. And in our statistical work, we had a lot of help from statisticians, and some of the computational modeling we got guidance to. And typical of all scholarly work, with every answer, more questions came to the surface, and surprise revelations took us on beautiful tangents that revealed new understanding. So what did we learn from studying ciprofloxacin and oxytetracycline sorption to 30 soils? And so I might say 30 soils, but these are hundreds of experiments to get these data points. So we identified that these soil properties, first we could identify the soil properties that were important or influenced sorption. So initially we measured ciprofloxacin sorption in this form when there was both a positive and a negative charge. And we found that surface area, or the texture of the soil, how much negative charge there were, how much iron and aluminum there was on the surface, all really influenced how much oxytetracycline sorbed. With ciprofloxacin, we got a little smarter and looked at the sorption when the compound was just positive and when it had both a positive and a negative charge. And in this case, we found largely the negative charge was what was important, but the surface iron and aluminum played a role when the pH of the surroundings was super high. Then what our statistical analysis told us was that these soil properties were important, and they were statistically significant in determining the sorption, but they were not good predictors. We could not predict or anticipate sorption based on these soil properties. And so we needed something else to do this prediction. But however, before we went into the prediction, we thought from these observations and the wealth of previous studies and a lot of outliers we dug into in our own data, we could understand the mechanism or the driving force or how these interactions took place. So for ciprofloxacin, we found that the primary driving force of the mechanism of absorption is something called cation exchange. To remember what a cation is, the T looks like a plus charge, and I learned this from my students, and I overheard them telling each other how to remember what a cation was, so I'm sharing full credit to my students. So a cation exchange, what I mean by that is here we have our soil with a negatively charged site with a positive sodium next to it. If we think about our ciprofloxacin molecule here, this positive charge is drawn to this negatively charged site, and the sodium will pop off. And so what you have is this attraction of the positively charged part of the ciprofloxacin to the negatively charged part of the molecule. So this was the primary mode in which ciprofloxacin was interacting with the surface, and in select circumstances, we saw that binding to iron or aluminum on the surface may be important. So if you look at it this way, here is the soil, and we'll watch the iron here. So if you have the ciprofloxacin molecule, this part of the molecule that can be negatively charged is drawn to the iron, and the OH group pops off. And so this is called surface complexation, where there's a bond formed between the negatively charged part of the compound and the iron here, okay? And so it was a little bit different for oxytetracycline, and we were a bit surprised by that. Oxytetracycline definitely sorbed by that first process, which I called cation exchange, with a positive ion was attracted to the negative surface. It was also drawn to the surface by this complexation between the oxygen and the iron. But as we followed the outliers, there was this other interaction we found. So a kind of bridging mechanism. So we're going to watch this negative charge site on the soil, which has aluminum with a +3 charge next to it. And the idea is that the negative part of oxytetracycline gets drawn to the positive charge. And so the positive charge that already exists on the soil next to the negative charge acts as a bridge to bind the compound to the soil here. And so what stuck with us is soils are an important filter for both these pharmaceuticals, particularly if the soil carries a negative charge, and a lot of soils do carry a good amount of negative charge. But what continued to perplex us is why these two compounds had different mechanisms. They are seemingly similar. They have a positive and a negative charge. Why are their driving forces different? Why is one predominantly drawn to a negative charge, whereas another is also binding to the iron and doing this bridging mechanism? We tried instrumentally to visualize the surface, but it was very complicated and we didn't get very far. The idea is because soil has multiple receptor sites where this compound can go, the negative sites, the positive sites, the superficial iron and aluminum. And what we've seen so far is these pharmaceuticals are ridiculously complex in that you can have a positively charged part of the compound interact with the negatively charged part of the soil, but you can also have the negatively charged part of the compound bridge onto a negative site with another naturally occurring ion there. Or you can have that same negatively charged part bind to iron on the surface. So there were too many mechanisms going on, too many modes of interactions to unpack. And this seemed, hard to really get more deeper as what's happening mechanistically. So in order to do that, we decided to complement our studies to look at substructures. So we started out really hard and we said, let's get simple. And we looked at the sorption of smaller chemicals that mimic the important part of these compounds. So we looked at a whole host of substructures of oxytetracycline, a whole host of substructures of ciprofloxacin, and we also measured their sorption both onto soils and pure phase minerals. So pure phase minerals are minerals extracted out of soil. So soils are super complex, and I showed you there are so many receptor sites. But if we go to pure phase minerals, this white powder is an aluminosilicate. It only has negative sites. This yellow powder is an iron oxide. It only has superficial iron. And I put in this picture of Goethe for the humanists in the crowd because this iron oxide is called goethite. And if you want to know why geoscientists name their oxide goethite, you can talk to Emily or Rachel, who are in the audience today. And so we looked at, now we went much simpler and said, let's look at small molecules and look at components of soil that are really simple to tease out what might be going on. And so we arrived at a couple of things after a lot of studies with these compounds, is that how much the charge was smeared was important. So if you think about, these are two molecular structures. They all just have a +1 charge. And the blue is the positive, and as you get negative, it's more red. So when the blue gets lighter, as you see here, the positive charge is more smeared out. And in this molecule, see, this is more blue here, but that blue is more smeared out. So think of like a blob of peanut butter versus smeared peanut butter, right? And so the charge is smeared like peanut butter over a larger surface area. And so the smearing, how spread the charge was on the molecule, plays a big role in the mechanism by which these compounds might be interacting. So for example, if the charge is smeared, the compound can lie down on a negatively charged surface to maximize how much positive charge can interact with the negative charge. If the compound did not have very much smeared charge, then the negative red becomes repulsive to a negative surface. So these compounds will tend to stand up and not lie down. And so they can maximize the attractive force between the positive and the negative and minimize the repulsive force between the negative and the negative. And what we also found that in addition to the smearing, what mattered was how close the positive and negative charge were to the surface. And so we come back to oxytetracycline and ciprofloxacin here. The ciprofloxacin negative charge and positive charge are very far away, whereas they're close by in oxytetracycline. So what ciprofloxacin can do is that its charge is not smeared, so it stands up straight, and the standing up straight helps because it keeps the negative part far away from the negative, and the repulsion is less. In the oxytetracycline, the positive is attracted to the negative surface, but it's not super welcoming because that other negative charge is right next door, and there's a repulsive interaction there. So we could see why now that oxytetracycline much preferred just the negative parts of the soil versus ciprofloxacin much preferred the negative parts of the soil, whereas oxytetracycline kind of did, but really preferred to bind iron or other sites on the soil because this repulsion hindered it moving to the negative soil. And so this work showed us that compounds really orient on the surface in a way they can maximize the attraction and minimize the repulsive forces. As we were figuring out our experimental conditions, we started seeing some outliers when we looked at sorption profiles. And there was this weird behavior. We started seeing sorption was increasing more than it should for the same compound when we were changing experimental conditions. So first we thought it's the instrument. Then it wasn't. Then we thought it was us. And we did it over and over again. And it was not us. It was not the instrument. There was something real. So then there was another journey with 8 substructures, several pure phase compounds, many, many experiments. And what we found was something really cool. What we found would happen is that a positively charged molecule will absorb to a negative surface like it did, as I told you before. But we found this other cool interaction wherein a second molecule would go in there even before all the sites were occupied. And this positive charge on this compound would interact with the ring of this compound. And this happened particularly when this charge was more smeared out and there was more negative charge there. So that was kind of a very cool interaction we found that we weren't expecting to find. We also found that in some cases, we saw sorption more than the number of receptor sites there could be, and that was interesting. And that was because these rings would stack up in a way that you could minimize the attraction, minimize the repulsion, and maximize the attraction. And so that was a really nice journey with outliers to get to what might be another additional interaction we found on the surface. So this was all quite cool. We had a better grasp on the mechanism now. We know what soil properties and receptor sites were important, but we could not predict yet, right? And the prediction is really important because it isn't feasible to measure the sorption of every soil pharmaceutical combination. Because we know the soil in your front yard is different from the soil in your backyard, and there's a whole host of measurements. It's not really realistic when we think about fate in the environment and we think about predicting and anticipating fate. So we started thinking, could we do some base characterization of soils and accommodate and allow for all of these things that we had already understood, that there were certain soil receptors that were important, certain chemical properties that were important, certain mechanisms of interaction that were important. And so what could we do? We had hit a wall using soil properties, and existing models for prediction only worked when the compound was interacting via one mechanism. And as we had found out, our pharmaceuticals sorbed to multiple sites by multiple modes of interaction. So at this point, Allison, my collaborator, and I took stock of our work together, the individual work in our labs, and decided we needed to leverage this mechanistic understanding that we had gained to propose a different way we could think about prediction. So we thought we'd propose this mechanistic framework, keeping in mind there are 5 receptor sites on soil, as I pointed out before: negative, positive, polar, the surface iron and aluminum, and these nonpolar domains. And keeping in mind that there's this complexity, there's not this one-to-one match. The same molecule can interact by 3 different mechanisms. One group can bind to 2 different sites. And how do we map this in a way that makes sense and we can move towards a predictive capability? So what we did is we thought about a mechanism-based model, and again, we went back to these smaller structures. And we said, can we create a mechanism-based model that maps onto soil receptor sites? So what we tried to do is to pick a small substructure, a small compound like this, and here that could actually probe the soil and tell us how many sites are relevant, because clearly the soil properties we normally use weren't working. So we picked 2 positively charged compounds as a positively charged compound probe. And instead of measuring soil properties, we measured how much sorbed, or we measured the KD for these probes by this cation exchange process, the positive attracting to the negative. So the idea was that if we measured how much stayed on the soil, that would tell us how much negative charge there was and what the driving force for sorption is. So we would get one step, the driving force, and in the next step, what we would do is we had to go from the simple compound back to our complex molecule. And our hope was to do that by creating what we call structural correction factors. This could be a library of of numbers that helped us scale up based on experiments that we would do or computational work we would do. So the idea is that you could predict how much of this compound sorbed if you knew how much the probe sorbed and you had a library of to-be-determined correction factors that could take us from here to there. So that was the idea behind the predictive model and how we could move there. So the next slide, just hang on tight. There's an equation. It's the only equation. And we'll get through it together, so don't lose me. So we can think about the total amount of a chemical that's sorbed as relevant to neutral molecules, which I'll call type 1, and we'll completely ignore this because the literature has, this is well established, so we don't need to go there. This next piece is for positively charged compounds that sorb via the cation exchange process. And the second is for compounds that can carry a negative charge that interact by two mechanisms, that bridging mechanism and the binding to iron and aluminum. So the idea was we could predict this. This is already in the literature, so we know how to do that piece. Then we could measure the receptor sites by using a probe, which means we have to find a probe. We could find another probe for compounds that carry negative charge, and then we develop scaling factors for the positively charged part and scaling factors for the negatively charged part. And so we have two separate ongoing projects in our lab, one focused on positively charged compounds and one focused on negatively charged compounds. So Team DIRT goes into Team Anion and Team Cation at this point. And with respect to the positively charged compounds, we studied two probes, these small molecules, PTMA and benzylamine. And the idea is that they're going to get at this exchange. They're going to quantify the negative charge on the soil. We also looked at two pharmaceuticals, Tramadol and Discipramine. So we measured their sorption onto 30 soils and some pure phase minerals. Allison's lab looked at 8 other pharmaceuticals and really focused on looking at pure phase minerals. And what we found was that PTMA turned out to be a good probe. So how did we do it? We literally did hundreds of sorption experiments and then ran a lot of correlational analysis. So what did we find? So I'm going to show you some graphs here. So the y-axis has our pharmaceutical, either Discipramine or Tramadol, and the y-axis is going to change in the 3 sets of graphs I'll show you. Shown here is the negative charge on the soil property. And so what we see here is we kind of have a moderate correlation. You want that number R squared to be close to 1, so you don't want to see so much scatter. You want to see that the lines sort of correlate and line up within those 2 bars. So there's a little bit spread out here. So we knew that the soil properties weren't doing a good job predicting. So we went to the next probe, which is benzylamine. It did a little bit better. We got a little bit a stronger correlation. So again, the measurement of how much benzylamine sorb is now on the y-axis, and the pharmaceuticals are still on the x-axis. And then we looked at PTMA. And here we see a strong correlation, like 0.9, very close to 1. So the correlational analysis says that the driving forces that get PTMA on the surface are very similar to the driving forces that are getting our pharmaceuticals on the surface. And so we could conclude that PTMA was a pretty effective probe for these positively charged molecules. And so we thought, okay, we have a handle on the probes. We kind of have that. What about the structural scaling factors there? And so what we started finding as we thought about the structural scaling factors is that in the literature, Droge and co-workers had sort of been able to predict for very simple pure phase solid material. They could predict sorption from compound size and the amount of smearing, or a surrogate for the amount of smearing of charge that could happen. So we took their model and adapted it to our probe idea, and we did pretty well. So here we see our experimental values on the y-axis and the predicted value using this on the, on the x-axis is the experimental value, and the predicted value is on the y-axis. And we do pretty well in terms of the correlation. So it seems like we have a good handle on what the probe should be and what the structural correction factor might be. So just when we were thinking, we got that, we started asking, what if the charge was even more smeared, right? So here is loratadine, which is found in Claritin. So the positive charge, when you put it in the ring, the smearing can be like a lot of smearing. The charge really spreads around that ring. And we started looking at substructures and thinking about what happens then with these molecules when it's even more smeared. So that's what my students are currently working on right now. And the last few years, we've been thinking about, does this model work when the charge is even more smeared? We're somewhat optimistic, but that PTMA might still be a good probe. But these molecules with the nitrogen within the molecule, the scaling factors, you know, the jury's out, particularly when the compound is very small. We're not sure that that scaling factor is going to do as well. So the fun continues, and there's more to learn about the positively charged prediction piece here. If we now move to the negatively charged compounds. And so if we think here, so we kind of have a handle on that. So what about here? What about compounds that carry a negative charge? So this is where team anion comes in. So anion meaning negatively charged compounds. And so essentially to remind you, if compounds can carry negative charge, they can complex to the iron and aluminum on the surface, or they can bridge via a bridging mechanism. So we spent a bunch of time on this, literally this time over 1,000 experiments, and the results are not so straightforward, but we've had a lot of fun and a lot of learning. So we looked at 3 pharmaceuticals, ibuprofen, which many people in this room have consumed, sulfasalazine, and carbapenicillin. So those were our pharmaceuticals we looked at. And we looked at 8 probe compounds, some with just 1 negative charge, 4 where the negative charge could be next to each other, and 2 where the negative charges were far apart, right? So this is, we studied them onto 30 soils, onto several pure phase minerals. And what we are arriving at is that a single negatively charged probe may not be viable, right? Particularly because there are 2 mechanisms by which these compounds can sorb. So what we found so far is that if you have 1 negatively charged group like ibuprofen, a probe with 1 group works well, very well to predict. If you have a compound with neighboring groups which can carry a negative charge, they do well for compounds like sulfasalazine that have a negative charge next to each other. When you have the negative charges far away, it's much more nuanced. It depends on the structure, it depends on the mechanism. And so where we're at with this is that while we were saying, okay, let's study a neutral compound that has groups, we again had a bunch of outliers and discovered yet another mechanism by which these compounds sorb. And we found that instead of the O groups, which could carry the negative charge interacting with the surface, we found that the ring could complex only to calcium, not to other ions on the soil. So this was an interesting discovery. So clearly, a single probe is not viable, and it has had us rethinking the model, really thinking about, do we need to have multiple probes, and how do we build in this nuance to be able to predict these compounds that can carry a negative charge? And so I think also coming back, how much can computational chemistry help us with some of this analysis? And then we haven't even started on the scaling factors, right? So the fun continues there. So I'm going to wrap up by saying that we have contributed to the understanding of various forces that drive pharmaceutical from water onto soils, and to help understand how soil encounters filter pharmaceuticals and influence the fate of pharmaceuticals in the environment. We may have made a little bit of a dent in terms of the predictive capability, but we really have a long way to go with predictive capability. And what I'd like to do is end with a lot of gratitude. First, to my parents, family, and friends. I'm so, so fortunate for all the love and support that I have. And thanks to my mother's collaborative work with Women in Maine, I had my Maine family set up for me even before I came to Bowdoin, and I am so delighted that 2 of them are in the audience today, and some of them are joining us via live stream. And to everyone in chemistry and ES, and now also in academic affairs, ever supportive, whether it's unexpectedly I have to travel over oceans or I needed to be reassured that this title of this talk would be okay. You've always had my back, thank you. And my undergraduate and graduate advisor, particularly my undergraduate advisor, who got me hooked to the connection between chemical structure and behavior. And before the Council of Mentors was a thing, I had my own council of senior women mentors at this college. As someone who went through STEM, undergrad and grad school, seeing very few female role models, I came to Bowdoin. I had senior women in all divisions of the college who were invested in my success, and it means so much to me that many of you are in the audience today, so thank you. And finally, I'm going to end where I began, with gratitude to my research students who brought me and bring me a lot of joy and laughter. Here's an example of them documenting a visit to LL Bean at 4 a.m. in tie-dye lab coats, and another demonstrating really weird notions of what hazardous waste might be. They have made me a better human, a better teacher, and a better scholar, and I'm really thankful for that. And I'm happy to take your questions. I've been asked because of the live stream, if you could come to the aisle mics to ask your questions, that would be very helpful. So thank you. Thank you. Thank you. Thanks.
Great talk, and we're just so happy to celebrate you tonight. I just want to ask a little bit about your tools. I was sort of sitting in the talk thinking about you like going around with a little spade and a bucket, collecting some soil, going to the pharmacy and asking for some of their drugs to bring back to your lab. Could you tell us a little bit about
absolutely.
sort of the process of collecting data?
Yes, happy to. So it's actually a good question. When I was in North Carolina at Duke, when we started collecting these samples, I was super naive, and I just thought I would go meet farmers and ask them for their soil or adjacent soil, because I wanted uncontaminated soil so I could contaminate it in the lab. And then I learned very quickly that farmers are very suspicious of scientists. So instead of me going, my lab tech and students went. And we had a lot of help from extension agents who actually gave us the soils from some of these parts where we didn't travel to. And so what you try to do is you try to get soil in one area, and then you really homogenize it so it's sort of representative of the soil. So there are 12 soil orders in the world, and Hawaii has all of them except gelisols, but that's just a nerdy soil thing. But so we were able to get soils from 5 orders in the US. That's why we picked the eastern United States, and as you go towards the south, you see more expanding soils, and so we really picked like 5 types of soils within each category. So literally, like, we have buckets of soils that we have, that we've characterized them so they've become precious, because we spent a lot of time and money figuring out all these, like, how to characterize these soils. And so, yeah, so the soils are there in these buckets, and then we weigh them into these little test tubes to do our experiments. In terms of the pharmaceuticals, you can buy them. Some of them you can buy just directly from Sigma Aldrich, purified in little jars. Some of them, I was very naive to Ren Bernier's chagrin. Some of them are controlled substances, so we had to be careful to decide which ones we could buy. And so, yeah, and the substructures are easier to buy, because you look at what substructure you want and you see if it's available, and you can buy it at Sigma Aldrich, which is like, you know, the I don't know, the Walmart of the chemical industry. Yeah. Yeah.
Thank you for the unsurprisingly great talk. I was wondering about your thoughts having to do with even beyond it's not complicated enough. So what about impacts of things like climate change or acid rain or salting roads or things of that sort on these properties? If the thought is that soils can be our friend by taking these out of the waters, could you talk a little bit about that?
Yeah, absolutely. What I didn't talk a lot about is soils also could be acidic or have a pH. So you just put the soil in water and then you measure the pH of the soil water. And in our lab, it so happens that some of the soils that we have been limed because they were used for agriculture. So as soils acidify, farmers line them to keep them at the right pH for growing materials. And so we already have some soils that are super acidic and some that are not acidic. And so, Anne, you ask a really important question. So if you have a lot of acid rain, soil charge can also change with pH. So I simplified and didn't really talk about this. And so what can happen is the acid can occupy and block some of these sites. And so the filtration capacity of the soil will become less and less, right? And in terms of temperatures, the sorption phenomena itself, there's not a huge effect that we see with temperature change. But it might change there will be some effect, but I think the numerical change in the numerical value is going to be within error for the time of few degrees change in temperature in the soil systems. Yeah, thank you. Yeah, Bruce.
Do you have enough examples yet to start to use machine learning? Because your possibilities of chemicals and soils is huge.
Yeah, thank you. So we've been working with a computational chemist at UConn, and on the chemical structure side, the computational piece is easy. On the soil side, it's really hard. So even with the computational work with minerals, we've had some success in predictive capability, but once you get to soils, it becomes really hard in the computational models to put it in water, and then that water signal is really hard. But we learn a lot from from biological systems, and some of the computational modeling work was mimicked on what's already used in biological systems, because you're dealing with the same hydrophobic charge, stacking kind of forces. It's the soil variability and heterogeneity piece that's really hindering. So we could do much better with these pure phase minerals, but harder with soils. Yeah, Matt.
This is adjusted for Bruce's height, not mine. That was great, Dharni. I listened to some of it while I was driving my daughter around, so I caught most of it. But thank you, and I'm glad to be here celebrating tonight. I have an environmental studies question for you, since you are the Stanley Druckenmiller Professor of Chemistry and Environmental Studies. The question I've got for you is, well, actually 2 parts. You talked a little bit about pharmaceuticals in the environment, but after they pass through the human body, we take our Tramadol for pain, we take our ibuprofen for being middle-aged, we take our Prozac or what have you. It passes through our system in ways we don't need to describe. And it gets in water systems and soils and places in pretty small amounts. We've only been able to measure it recently. So first of all, why should we be concerned, and speaking as a chemist as well as an environmental studies scholar, about very small concentrations, relatively speaking, of things like pharmaceuticals in the environment? And secondly, since it is so small, and if there is a reason for concern, what are the challenges once you figure out with your colleagues how to measure them accurately? What are the challenges then trying to help prevent them from getting into the environment, since most of our wastewater is designed at removing the ways in which those things get into the environment, or poop and pee, but not so much other things?
Yeah, no, so thank you. So yeah, an obvious question is we consume this stuff, so why should we be worried, right? Like we literally eat giant amounts of them. The other, and so the idea is why, and the reason is antibiotic resistance, particularly for antibiotic pharmaceuticals. So if there's a constant amount, even a low amount, always present, our microbes are adapting, so they become resistant to these to the pharmaceuticals, and so then we have to keep discovering new pharmaceuticals which might not be viable. And so the motivation to really understand what's happening to pharmaceuticals came a big piece of it was from antibiotic resistance. The other thing is ecosystem effects that we don't know. So Prozac is really affecting fish, for example, in ecosystems. So pharmaceuticals can have effect on organisms that are very different than the effect it can have on us, right? And there was a last piece to that question.
Regulation. I mean, once we measure, how do we regulate it?
So
we're taking them, and wastewater doesn't remove this.
Right. So yeah, so the wastewater is designed to basically get rid of organic matter, which is poop and pee, right? And so these compounds, as I said, have charge. They like to stay in the water. They're not going to bind our treatment systems really well. And so some of the other research is going on is like, can we use UV treatment to break them down in wastewater treatment plants? The real worry is, will UV then treatment and breakdown create other substructures that might have other unknown toxicities that we don't know, right? And so it doesn't mean half the chemical will be less toxic or might not have another effect. So more of the work is going towards remediation and breakdown there. Yeah.
Dharni, thank you so much for a great talk. For my question, I want to reference one of your colleagues, Danielle Dubey, who's one of her advisors, as many of us know, recently was awarded a Nobel Prize. And in her remarks afterward, she talked about Danielle and others, and she talked about the importance of diversity in the lab and what it means to have people from different backgrounds together, asking different kinds of questions, bringing their life experiences, et cetera. And I wonder if you could talk about that in your work.
Yeah. So first I wanted to say how excited I was when Carolyn Bertozzi won the Nobel, because we've heard so much from Danielle, and just, I was spamming everyone in our office with like short videos or whatever I could find of really how she's such a model for thinking of inclusive excellence and diverse perspectives. I feel I've really seen it in my lab. My students will take Ben's class and come back and say, I think I figured out how this molecule is going to orient on the surface. It's something I would have never thought of because, and you just see that you have different voices bringing different ideas in, right? And just sort of the excitement. Like Seamus, who's in the audience, had to contain him from saying let's go bother Emily and Rachel and try to find out what's going to happen here. And we're like, yes, we'll do that after we find this out. But these rich questions, or Mariah saying let's change the pH, then we'll block out one site and measure another site. And so you just see different students, different people bringing in different ideas to the table that really moves the needle. And so this idea that people with different backgrounds thinking differently with different stories, and this just blows up the idea of a scientist has to be one way, right? And really the science is richer for the diversity we bring to science, our perspectives, our life stories. And And my students teach me that every day. So it's, yeah, it's amazing. All right, I think, well, thank you for being here and being here this late. I'm really honored. Thank you so much.